Hello, my lovely anatomists and physiologists. We are ready to investigate the kidneys and nephron in a little bit more detail. Remember when we talked about the nephron, we said it's the functional unit of the kidney. So we cannot understand the kidney without understanding the nephron. And we're going to start with the start of the nephron. And the start of the nephron has two names. It can be called either Bowman's capsule or the glomerular capsule. And we'll see that the job of the Bowman's capsule or glomerular capsule is filtration. And when we talk about filtration, remember, we're going to see that pressure will force fluid in solutes across a semi-permeable membrane. And the movement is based on size. So stuff that is able to actually cross based on the size of the openings will cross. So there's not a selectivity based on anything other than size. When we're talking here about filtration, we're going to see that blood will move through the capsule and we'll see 10 to 20% of the plasma will enter what's going to be called the capsular space. So we'll look at a model here in just a few minutes to help us visualize what's going on here. And when we're talking about material moving, we're going to see, again, it's indiscriminate. So we're going to have, for fluid and solutes, we're talking water, we're talking nutrients. So nutrients would be amino acids, would be glucose, would be vitamins. So stuff we don't actually want to be present in the urine will actually leave here. We'll have electrolytes leaving. So there's really, you should think like a pretty big exit of material actually leaving at the Bowman's or glomerular capsule. Now, the good news is the fluid doesn't stop here. So it's going to go through the rest of the nephron. And so the job of the other parts of the nephron will be to sort of fine tune, making like the perfect urine for the body at that point in time. So let's take a look at the Bowman's capsule or glomerular capsule. And what we should see is that the glomerular capsule is going to encase what's called the glomerulus. And so what we're looking at in this model is a cut through Bowman's capsule. And we're able to see inside the capsule. So it's going to be like a circular shaped structure, and inside it has a whole mass of capillaries. And the mass of capillaries is called the glomerulus. And if you notice on this diagram, we can see its texture. There's a little pinprick looking texture to the glomerulus. And so we're seeing that it is a fenestrated capillary. And we know from our study of cardiovascular system, then this is going to allow for a lot of movement across from the bloodstream into this capsular space. So this space that we're going to see, we're going to call the capsular space. Now, if we go back to our picture of the glomerulus, notice there's going to be an entry and an exit. So we're going to see the afferent arteriole as the entry. So let's draw movement in. And then we're going to have the efferent arteriole as the exit. And notice that the afferent arteriole has a large opening and a larger diameter, whereas the efferent has a smaller exit, smaller diameter. And that will be really important when we talk about the blood pressure and regulating the blood pressure inside the glomerulus. So then the kidney tissue that's encasing the glomerulus, that's what we're calling our Bowman's capsule or glomerular capsule. But notice that you have these kidney cells that are forming the capsule, but also like actually wrapping super tight around the glomerulus. So if we go back to our discussion of the Bowman's capsule, we'll see that we're going to have what we can call the parietal layer. So we'll have our parietal layer, which we know is the outer layer. And this is going to be composed of simple squamous epithelial tissue. And then we're going to have the visceral layer. And visceral, you know, remember I've said like all along, think of guts. So this is closest to the internal organs. In this case, this is going to be the innermost. This is going to be wrapping around the glomerulus. And we have a special type of cell here that we call a podocyte. Now the podocyte is going to have these like feet-like extensions that will actually like hug and wrap around the glomerulus, around those capillaries. So what we're looking at here, this group of cells immediately on top of the glomerulus, this is our podocyte. And we see that it has these extensions of the cell that will actually wrap around that glomerulus. But then we'll have these like spaces, and it looks like this little squiggly line where we can see the red peeking up through. And we're going to talk about these spaces here as a filtration slit. So we're going to see these podocytes are going to form the filtration slit. So that when we talk about filtration happening, we're going to see materials exiting the fenestrated. So the semi-permeable membrane that we're talking about is going to be the fenestrated glomerulus. And then it's going to go through the filtration slit. And so there's actually not a whole lot of regulation on the material that's actually leaving the glomerulus, which is how we're getting like 10 to 20% of the blood plasma actually exiting the glomerulus and going into that capsular space, and why we're getting stuff into the capsular space that, you know, we don't ultimately want to exit the body, things like nutrients, vitamins, right? Water. Taking a look back at this particular image, so here we're seeing the Bowman's capsule, the outer layer, that parietal layer here. You can tell that that's the simple squamous epithelium. And then we have here our visceral layer, which is composed of those podocytes. We're also able to see here that our fluid is going to move from the capsular space into what we're going to be calling the proximal convoluted tubule. So let's go ahead and label that as this sort of exit, and I'll draw an arrow that way. And then we see actually twisting around right up close to both the afferent and efferent arteriole, we have the distal convoluted tubule. Notice that in the proximal convoluted tubule, we have we'll come back to this, but we have cuboidal cells with microvilli. And so here is another example of a brush border. When we look at the distal convoluted tubule, we will see again a simple cuboidal, but it's lacking the microvilli. So that's going to be an important feature that we'll talk about here. When we talk about both the glomerulus, which is the capillary bed, and let's go ahead and call it Bowman's capsule, we'll talk about all of this as being the renal corpuscle. So the glomerular capsule is the actual kidney tissue, or we can call it the Bowman's capsule. The glomerulus is the fenestrated capillary that's inside. And then that whole collection of tissue is referred to as the renal corpuscle. So this is really like our starting point for making our urine. When we're looking at that visceral layer, and we talked about it as like intimately hugging around the glomerulus, and we said it was composed of those podocytes, which are important in forming the filtration slit. We'll also see a type of cell here called the mesangial cell. And the mesangial cell can actually contract and help regulate filtration. And we'll see it's doing that because it's going to, I'm going to put this in parentheses, help regulate what we're going to call glomerular filtration. No, excuse me, glomerular hydrostatic pressure. So we'll talk more about this in the future, but we'll see that the mesangial cell is going to be involved in actually helping to regulate the glomerular hydrostatic pressure by actually contracting. It's going to be positioned in such that it's causing a little bit of constriction of the glomerulus, essentially. If we go back to this diagram, remember we're seeing the afferent arteriole entering into the glomerulus. The glomerulus is surrounded by the Bowman's capsule. We see the blood is exiting out of the efferent arteriole. We can see the visceral layer, those podocytes firmly wrapping around the glomerulus. And then we have, of course, that capsular space in between the glomerulus and the Bowman's capsule. We've labeled the exit of the capsule as the proximal convoluted tubule. And then we've said folded up, coming around in between the afferent and efferent arterioles, we see the distal convoluted tubule. We also have here what's described as the juxtaglomular apparatus. So we have a group of cells, juxtaglomular apparatus, and we'll see in our OpenStax book, they'll abbreviate this JGA. Okay, so let's go back up to our notes on Bowman's capsule. And let's talk a little bit more about the JGA. JGA, Juxtaglomular Apparatus. Juxta, remember, means like close to. So these cells are close to the glomerular capsule and the glomerulus. Now we're going to see that these are basically like, I'm going to just label them here, your endocrine cells. So when we talk about the job of the kidney, we've mentioned like it's producing 80% of the erythropoietin. So we're seeing that's happening from this group of cells here. We'll see that we will have in the JGA the macula densa. Macula densa is going to help to regulate fluid composition. Of the distal convoluted tubule, which we can abbreviate DCT. And so there is going to be looking both at the concentration of sodium ion and at the flow rate. So the macula densa is helping to regulate fluid composition of the distal convoluted tubule by helping to monitor sodium ion concentration levels as well as the rate of flow. We'll see that these cells can release both ATP and adenosine, which will act locally. And we'll see this as part of like auto-regulation. So we'll come back and talk about this a little bit more in the future. The macula densa also is going to release renin. So we're going to talk about this quite a bit. We talked about it quite a lot before in the cardiovascular system, but we're going to come back to this release of renin, which leads to activation of angiotensin 1 and 2, which leads to the release of both aldosterone and ADH. So we'll come back to that. We also have, as part of the juxtaglomular apparatus, a group of cells called juxtaglomular cells. And the juxtaglomular cells are going to be actually smooth muscle. Of that afferent arteriole, which can either constrict or dilate as needed. So we'll see one of the major ways that we can control filtration that's happening at the glomerular capsule is by controlling the diameter of the afferent arteriole. So we'll come back to that idea, I think, as we go through. Okay. Up next, let's take a look at the details of that glomerular hydrostatic pressure and how we actually get filtration to occur. So stay tuned. As always, take care of yourselves and each other.